listen guys, yes. And today we'll be learning about DNA. Specifically, replication. So DNA is short for deoxyribonucleic acid. DNA has a sugar phosphate backbone with C paired to G and A paired to T. DNA strands are coiled around a histone and supercoiled to make a chromosome. Chromosomes hold all the genetic material in them. Cells are always replicating, and you have 23 pairs of these in every single cell.
Wow.
So, how do they replicate?
I don't know.
We're about to find out.
I'm sure if any of you have ever taken a rope and twisted it, like the double helical nature of DNA, and started to pull it apart, you'll find that the rope starts to coil in itself, like this. It starts to get tense and eventually will fold back on itself. This makes it difficult and near impossible for DNA to replicate, but this is supercoiling, and it's in fact necessary for the DNA process. However, it's not necessarily for replication. In replication, we have an enzyme called topoisomerase, which looks like this. So, how topoisomerase works is it comes into this supercoiled DNA strand, and it will make an incision, and then with that incision, it will unwind one of the twists here, which relieves the tension and strain in the strand. So it can continue replicating, and it will unfold on itself and turn back into the strand that it was meant to be. And this relieved tension is how DNA replicates when it keeps on pulling apart.
DNA replication starts at the origin of replication. The origin of replication will form at specific sites where two parental strands will separate to form
the DNA replication bubble.
To form a DNA replication bubble, helicase, an enzyme, will cut the DNA strand in half, starting at the replication fork. Not that kind of fork. The replication fork is the Y-shaped area on the ends of replication bubbles. Now the parental strands are going to serve as templates for the new daughter strands.
DNA replicates from the 5th carbon to the 3rd carbon because nucleotides can only be added to the free 3rd carbon end of DNA. This is also known as the 5' to 3' direction.
In this half of a DNA replication bubble, you see lagging strand and leading strand,
which are
the daughter strands. The leading strand replicates continuously towards the replication fork. The lagging strand progresses in a series of segments away from the replication fork,
but still in the mandatory 5' to 3' direction.
After helicase, it splits the parental DNA strand into 2 template strands. Single binding proteins will come and stabilize the singular base pairs until DNA polymerase III adds the complementary base pairs to make DNA strands.
DNA replication is slower on the lagging strand than on the leading strand by 1,000th of a second, because the origin of replication begins on the leading strand.
The leading strand is faster in replication than the lagging strand because DNA polymerase III can work continuously toward the replication fork only in the leading strand, while in the lagging strand, it must proceed away from the replication fork.
DNA polymerase III synthesizes the leading strand continuously in the 5' to 3' direction. It also adds onto the primer in the leading strand and the multiple primers in the lagging strands.
Primase is an RNA enzyme that joins nucleotides of RNA together into a primer. The primer can be made up of 5 to 10 nucleotides. The primer initiates a nucleotide chain and is complementary to the template strand. See here, 5 to 3 and 3 to 5. This is the lagging strand, and this is the primer that will come and attach to it. The base pairs are aligned so that the hydroxyl group joins at the 3' end of the next nucleotide, and it continues along the rest of the primer. Once the primer ends, DNA polymerase III will come in and add more nucleotides to the 3' end.
The lagging strand has many primers needed to prime synthesis for Okazaki fragments. Okazaki fragments are called Okazaki fragments because a man named Okazaki discovered Okazaki fragments. What causes Okazaki fragments, you might ask? Well, as the lagging strand's replication bubble opens, DNA polymerase III attaches its template, which synthesizes a short strand of DNA. The bubble will continue to grow, with more segments being added in a similar fashion.
Alright class, welcome to Loyd's biology class. So here you can see that we've got the lagging strand of DNA, which has already been split and primed by primase. So here you have your RNA primers, which are every 100 nucleotides, and each of them are 5 to 10 nucleotides long. It's kind of variable here. So we're going to zoom in here on this little section. So here in this section, you've got your adenine, and then you've got your 100 nucleotides of adenine. These box ones represent the 5 to 10, by the way. So what first is going to happen is DNA polymerase III is going to come jiggling along from the 5 to 3 on the top part. Then it's going to replace all of this nothing here with something. See, we're going to get DNA going in the opposite direction, anti-parallel, as you may very well know. So you're going to have all your little T's. So now that DNA polymerase III has come chugging along and done its job in this particular 2 sections, what's going to happen is now that this is filled, as you can see, it's not entirely DNA. We've got a very interesting composite of RNA primers and then DNA.
DNA polymerase I now comes in and fills these RNA primers with chunks of DNA to match the bottom. This is the end of the role of DNA polymerase 1 and 3 in this section. Afterwards, these will be finished and connected by ligase.
Okay, so DNA ligase, right here, links the Okazaki fragments, these guys right here, together with a phosphodiester bond, completing this section of DNA on the lagging strand. Restriction enzymes, which cut apart the DNA fragments, are like scissors, so the DNA ligase is like glue, connecting the fragments together again.
So how the DNA ligase connects the Okazaki fragments is by coming in and catalyzing the phosphate group on 5' and hydroxyl group on 3'. By doing this, it will create a sugar phosphate backbone.
Now, you might be wondering, wouldn't we eventually run out? Wouldn't there be an end replication problem? And to that, I answer you one solution, one hero, present specifically in eukaryotic cells, because many other kinds have circular DNA, which has no ends, and that cap on the end of our strands of DNA keeps our genes from being used up. That savior is a telomere. The sequence TTA GGG, repeated over and over again at the ends of all of your chromosomes, helps keep them from fusing together, causing chaos within your bodily systems, and also keeps them from running out too quickly. But telomeres can only do so much as they shorten and shorten as life goes on. A supporter of this brave hero, telomerase, an enzyme which can help lengthen yet again so that we do not die shortly after birth. In fact, with telomerase and telomeres, we can tell approximately how long somebody has lived, and it is one of the main contributing factors to death, because eventually you will run out of genetic material to copy, and telomeres can only do so much to protect you at the ends of your